really disgusting.
Okay, if I could have everybody's attention. It's almost 11 o'clock, so why don't we get started? It's my pleasure today to introduce our speaker, Dr. Cesar Rodriguez. Dr. Rodriguez received his Bachelor's of Science degree in biology from the Universidad Nacional Agraria. Did I get close on that? Okay, good. Yeah, he then went on to get his master's degree in entomology at Oregon State University, followed by a PhD in entomology at UC Riverside. He joined our faculty in 2005 to take on the responsibilities of an extension specialist in entomology with in the areas of blueberry and cranberry IPM, and also to develop a research program in chemical ecology, and he's going to talk about some of that research program with us today. So Cesar, I'm going to turn it over to you. You just need to share your screen and be all set.
Thank you, wow. What do I do here?
My ear really hurts.
I almost feel like I could fit my finger in your
Okay.
Okay, great. Thank you for that introduction. And as Dr. Hamilton said, I'm going to be talking about some of the work that we have been doing in the last 10 years to study the chemical ecology of fairly recent invasive pest, the spotted-wing Drosophila, Drosophila suzuki. So first I want to start by acknowledging all the people that from my lab that have been involved in this work. It has been, as I said, 10 years of work, and as you can see, there's a lot of undergraduates and grad students that were involved in this work, and they all came from different backgrounds and from different countries. And also, there's several postdocs that have been involved in this work, and again, coming from different backgrounds and countries. And this has been one of the main goals of my research, is to promote diversity within my lab. So I'm always happy to bring students and postdocs, early career professionals from different parts of the world. So I don't have to convince hopefully this group that invasive pests can be a bad thing. They can, there's an estimated amount of more than $1 trillion for the past 50 years on the cost, the economic cost that they have caused with these biological invasions. And also, this trend does not seem to end. As you can see in that graph, the projections seem to be increasing with more and more invasive pests arriving to different places. And one of the most places that have been most at risk have been the developing countries, including the US. As you can see there, I think the pointer doesn't work. As you can see there, including also New Jersey. Yes?
Sorry, they see in the presenter mode online.
Am I in the presenter mode?
Can I change it?
Oh, yes.
Yeah, go ahead. You'll be
Sorry.
Um,
I don't know how I've become the go-to technical person that people text when.
Go slowly.
Okay, I think that should be right. Those people online, if you can let us know. Is that okay? Great.
Okay, perfect. Thank you. So as I was saying, there's some areas in the world that are more at risk, and these are the developing countries, most likely because of increasing global trade. And, or now this doesn't work. do I?
Use the arrow keys.
No, doesn't work either.
I'll bring back.
Okay, now it works. Okay, let's continue. So, one such invasive pest is the spotted wing Drosophila, Drosophila susukii, and this is a fairly new invasive pest, as I mentioned. It came from Asia. It was first detected in the continental United States in California in 2008, and in our region in the Northeast in 2011. It has multiple hosts, so it attacks a lot of thin-skinned, soft, small fruits such as blueberries, raspberries, blackberries, cherries, strawberries. And they do that because the females have a prominent serrated ovipositor that allows them to attack ripening fruit, unlike other Drosophila species that attack fermented fruit, already rotten fruit. This pest has quickly invaded many parts of the world. You can see that it's already present in the North and South America, in Europe, and also in parts of Asia. So this is the outline of my talk today. I'm going to start by briefly introducing the crop, the blueberries. And then I'm going to talk about blueberry IPM and how this was disrupted with the arrival of the spotted wing Drosophila. I'm going to talk about spotted wing Drosophila itself and our work in looking at how we can control this pest using chemical ecology at multiple spatial scales, from the fruit level to the landscape level. I'm going to end with some conclusions and some future perspectives. So blueberries is an important crop, especially here in New Jersey, because here is it's where domesticated blueberries were originated. This is a native crop from the northern part of North America, from the US and Canada, and is one of only 3 cultivated crops that are native to the US. These include blueberries, cranberries, and Concord grapes. So because of that, United States is the world's largest producer of blueberries, and this is a very important berry crop in the US. It's second to strawberries, and the industry, the value of the industry is about $1 billion. And one of the reasons why we as humans consume blueberries is because of their health benefits. They have a lot of high levels of antioxidants that are good for human health. So blueberry production has been increasing throughout the last 2 decades. You can see that production of blueberries has been growing in Mexico, parts of South America like Peru, parts also from Asia, in Europe, parts of Africa, and in Australia. In New Jersey, blueberries, we grow only high bush blueberries, as opposed to, for example, Maine that produces low bush blueberries. And the main area of production is in the southern parts of the state, in Atlantic and Burlington counties, where blueberries are under production in over 9,000 acres. So New Jersey is currently the 6th highest blueberry producer in the US, after Oregon, Washington, California, Georgia, and Michigan. So the location where blueberries are produced is mainly in the Pine Barrens or Pinelands, which is in the southern part of the state. And here in our state, both blueberries and cultivated berries Wild and cultivated berries grow side by side in this region, in the pinelands. You have the pine forest, and in the understory you have a lot of wild blueberries that are grown there. And this is where, also in New Jersey, where blueberries were first domesticated. They were domesticated about 100 years ago by a person whose name was Elizabeth White. She was the daughter of a cranberry grower. So blueberries are attacked by about 16 insect and mite pests throughout the season. However, blueberry maggot fly was the main target of insecticide applications historically, before spotted wing Drosophila was introduced. And an IPM program was developed to control this pest. This is a native pest of blueberries, of this region that attacks blueberries. So we worked on developing monitoring tools for this pest, also to develop the treatment thresholds. Also, my lab worked in the development of reduced risk chemical control tactics to manage this pest, and also site-specific control management tactics. So the grower will just treat fields that were more at risk of infestation by this pest. So for instance, we did a study, an intensive monitoring study for blueberry maggot, where we placed traps throughout the southern part of the state in several farms. And we monitor the number of blueberry maggot flies captured in these traps. And these traps were located both along the edges of the fields and also in the interior of the fields. And this was done in 9 different farms. And what we found was that there was a significant effect of where those traps were located. We caught significantly more flies in traps that were closer to the forest than in the interior parts of the fields, indicating that these flies were coming from the forests adjacent to the blueberry fields. So the risk of infestation was higher if those fields were close to wooded areas. And this information was provided to the growers, and they used this information to apply insecticides only on those fields that were more at risk of infestation by this pest. So we were able to reduce the amount of insecticides that was being used to control this pest that was the driver of insecticide applications in blueberries in our state. However, this all changed when spotted wing Drosophila invaded us. So in 2011, spotted wing Drosophila came, and there were a lot of changes that happened after its arrival. So in collaboration with James Shopey from the Department of Environmental Sciences, we have been tracking historically the abundance of spotted wing Drosophila maggot and spotted wing Drosophila throughout the years. And as you can see in the graph, we're finding that the abundance of spotted wing Drosophila has been increasing since its invasion, whereas the numbers of blueberry maggot fly numbers have been in decline since spotted wing Drosophila invaded us. What was also very interesting is that if you look at the first appearance of both blueberry maggot fly and spotted wing Drosophila, spotted wing Drosophila has been appearing since the invasion earlier and earlier each year, whereas the blueberry maggot fly has been arriving later and later each year. This is a potential evidence for competitive displacement, where spotted wing Drosophila has been displacing this native pest, the spotted wing, the blueberry maggot. So another impact that it had was in the number of insecticide applications. Since invasion of this pest in 2011, there has been an increase in the number of sprays, insecticide sprays, that have been done in blueberry farms. So now growers have to do weekly sprays to control this pest. and the number of organophosphates, carbamates, pyrethroids, and spinosins has increased since this invasion. Whereas the amounts of neonicotinoids were effective against blueberry maggot, but not effective against spotted wing Drosophila, have been in decline. So, as you can imagine, blueberry farms are very variable in their landscape. And this is a landscape pest. There's a lot of hosts that it can attack that are present not just inside the fields, but also outside the fields. So we need to study the ecology, especially the chemical ecology of this pest at multiple spatial scales, from the landscape, from the fruit through the landscape. So I'm going to start by telling you some of the work that we have been doing to look at the chemical ecology of these interactions between this pest and the fruit. So one of the things that we quickly realized is that we were using monitoring tools based on fermented fruits to attract this fly. And we know that females are using ripening fruit, healthy fruit, to lay their eggs. But we didn't know much at the time on what were the chemical cues that they were using to find these hosts. So we did some studies looking at the volatiles emitted by the fruits and how they affect the behavior of this fly. So we looked at volatiles from blueberries, strawberries, raspberries, and cherries. What we did, we did an extract of these volatiles and conducted some olfactometer studies to look at the response of the flies to these volatiles. So what we found was that all the extracts were attractive. However, there were differences in how attractive they were to spotted wing Drosophila. With raspberries, raspberry volatiles always being more attractive to both males and females than, for example, cherry volatiles. We also did a 4-choice study where we exposed the flies to the 4 extracts at the same time. And we did that for males and females. And again, we found that both were consistently more attracted to the raspberry volatiles than to, for example, the cherry and the blueberry volatiles. So it was clear that there was something in these raspberry volatiles that were attractive to spotted wing Drosophila. So we wanted to identify what were those compounds that could be that were attractive. So in collaboration with Aijen Zhang from the USDA lab in Maryland, we did some couple gas chromatography and electron-antennogram detection studies to look at the antennal response of the flies to these to the raspberry blend. So this shows you the antennal response, the EAD response of the flies to the different compounds that are present in the raspberry extract. From these studies, we found that there were 11 compounds in that raspberry extract that consistently elicited a response, an antennal response in the flies. We also did a synthetic blend. We created a synthetic blend based on the natural blend and tested it on the insect antenna and found very similar responses. So what we did is we follow up these studies by creating a lure, a blend, a synthetic blend with these 11 compounds that were emitted by the raspberries. And we had these choice arenas where we compared the attraction of flies to the blend versus a blank. And we found that under laboratory conditions, the flies were highly attracted to this 11-component blend that we created. However, these traits that are used by spotted wing Drosophila to locate and utilize hosts can be influenced by humans, by the process of domestication. When we domesticate plants, we change the traits in plants, and these differences in these changes in these traits can affect the performance and preference of herbivores. So we were interested in understanding what are the consequences of domestication on fruit susceptibility to this invasive pest. So we know crops have been domesticated for about 10,000 years. And through that process, humans have selected traits that are important to us. And one of those traits have been the size of the fruiting organ. So for example, here I'm showing you the difference in size between the maize and its ancestor Teosinte. And one of we have evidence showing that by this process of domestication, we have made plants more susceptible to herbivory. So herbivores prefer or do better on plants that have been domesticated compared to their wild ancestor. Most likely because we have reduced the defenses that the plants used in the wild to protect themselves against the herbivores. So we wanted to ask that same question, especially considering that blueberries were domesticated here in New Jersey, and considering also that the process of domestication has been fairly short. And blueberries have been domesticated mainly for high fruit size and also high reproduction, which could have consequences on how susceptible they become to pests, especially to invasive pests. So we predicted that domesticated and wild blueberries would differ in their susceptibility and attraction to this newly invasive pest, Spotted-winged Drosophila. So for this, what we did was, for 2 years, we went out and collected fruit from both wild and cultivated sites. So we had these pair sites in all these different locations, and we collected both cultivated and wild fruits. And we conducted single choice assays where we exposed the fruit to a Spotted-winged Drosophila and look at their performance. So one of the things that was clear was that females prefer to lay eggs on the cultivated berries. So there were twice as many eggs on cultivated plants, fruits compared to the wild fruits.
Is that because of the volume of the domesticated?
Let's get back.
Yes.
Ask me that question later.
Okay.
I think so. Okay, so we also looked at the emergence. So there were a higher number of adults that emerged from the cultivated fruit than from the wild fruit. And yes, it could be because of the volume. But there were also different other traits that could have contributed to these differences. For example, the amounts of sugars are different. Wild blueberries have higher quantities of sugars compared to domesticated berries. They also have lower pH, so they are more sour. And also they have higher amounts of defensive compounds, such as anthocyanins and phenolics. Anthocyanins also give the color of the berries, so the wild blueberries tend to be darker than the cultivated berries. So we found that these domestic blueberries are larger and firmer, which could explain why they are more susceptible, especially the size. However, the wild berries are sweeter, more sour, better defended, and also darker in color. So we also wanted to see whether there was an effect in attraction, so whether there's a behavioral effect of domestication of blueberries on this fly. So we did similar studies, subsequent studies, where we went out to these similar sites. In this case, we only picked like 3 sites where we collected fruit from wild and cultivated plants. You can see that we bagged the fruits, so we prevented the fruits from being exposed to herbivores in the field, and also prevented from being sprayed by insecticides, because that could definitely affect their behavior and performance. So we brought the berries to the lab, and we conducted choice studies where we gave flies a choice between the wild and the cultivated berries. And we found consistently that the flies were more attractive to volatiles from the wild berries compared to the cultivated berries. So we did similar studies as I showed you before, where we looked at the compounds that were emitted by these wild berries that might be responsible for the attraction, and in collaboration with Agent Zhang, we identify those compounds. So first we found that the blends of emitted from wild and cultivated berries were different, as you can see in this graph. So each dot indicates a blend. So you can see that the wild berries produce a blend that is different from the domesticated one. And you can see that the blends from wild berries are more variable, which you would expect also. What we found doing GCEADs is that there were 9 compounds that consistently elicited an antennal response in the flies. These are from the wild berries. So we tested each of these compounds doing single choice assays, and we found that they were all attractive to spotted wing Drosophila. However, we found that 4 of them were most attractive. So we wanted to see whether we created a 4 blend and a 9 blend with these volatiles and tested them individually and as a choice test on the attraction of spotted wing Drosophila. So here is as a single choice, and also we tested it as a dual choice. So we found that both the 4 and the 9 component blends were attractive to spotted wing Drosophila, but when we compare them together, we found that they prefer the 4 component blend than the 9 component blend, indicating that some of the components in the 9 component blend were redundant. And this was the same case for mated females. We found the same effect with virgin females, and we also tested males, and we found the same effect. So both, regardless of sex and physiological state, we find that these flies are attractive to volatiles emitted from these wild berries. So what we find so far is that wild fruit produce higher volatile emissions and also higher chemical defenses. So they have more chemicals in a small size. And for that reason, they are more attractive by weight and also less susceptible than the cultivated blueberries that have lower emissions of volatiles and also lower chemical defenses. And thus, they are less attractive and more susceptible. So spotted wing Drosophila is clearly that it has a negative effect on the fruit. So if you have infestation, you're going to reduce the fruit that can be harvested. So it's bad for the blueberries and it's bad for the growers. But also there's microbes that interact with the fruit. However, how these microbes interact with the spotted wing Drosophila is not very well known. And it's known that some microbes can have a positive effect on a spotted wing Drosophila. For example, there's yeast that are symbiotic to spotted wing Drosophila that a spotted wing Drosophila uses to locate the fruit and also for larval development. They use it as protein for the larvae to develop in the fruit. But also we know that there can be microbes that can be have a negative effect on the spotted wing Drosophila. For example, a study showed that gray mold, Botrytis cinerea, infection by this mold, this is a mold that causes reduce in yields in raspberries. Infection of this mold repels spotted wing Drosophila, also reduces oviposition, and also reduces larval development. So it has a negative effect. Infection by this fungus has a negative effect on the performance and preference of this fly. So we wanted to look at how anthracnose, the most common fungal pathogen in blueberries, affects the preference and performance of the spotted wing Drosophila. So we asked, does anthracnose infection affects this invasive pest? So first we conducted studies looking at repellency, like whether this fungus repels flies. And we tested a solution that we sprayed on the fruit that will contain the spores, either spores from the colony, a lab colony, or collected from infected plants, fruits, and also a solution that had no spores. And we found that for females, the females were repelled by these solutions, fruits that were sprayed with solutions that contain the spores. So the spores from these microbes change something in the fruit that repels these flies. We also looked at the effect on males, and we found no effect of these solutions on the males. And this makes sense because females are the ones that are looking at these fruits as oviposition sites. So we also found that the number of eggs that the females lay on the infected fruit was lower. And also the number of adults that emerged from this infected fruit was reduced. And the performance of the larvae in the fruit was also negatively affected by the fungal infection. So subsequent studies done in collaboration with Caitlin Rehring from the USDA lab in Florida, and also work that my student Amanda Quadrell is doing, and she presented at the colloquium, is looking at what are the compounds in these infected anthracnose infected berries that are repelling spotted wing Drosophila. So what we did first was we looked at the volatile emissions, and we found that infected berries differ in the volatile emissions compared to the uninfected healthy berries. Next, we looked at, we identified 14 compounds that were differentially emitted by the infected berries. So we did a multiple choice assay to look at the effect of all these compounds on spotted wing Drosophila, and we found 9 compounds that that repelled the flies compared to the control. So we conducted further studies doing 2 choice assays, and we found that all of these 9 compounds repelled the spotted wing Drosophila. However, 2 of them caused more than 90% repellency. And this is something that Amanda will be continuing to look at. She's going to be looking at those responses, the effect of those on the repellency of these compounds on spotted wing Drosophila. And also she will be testing these compounds under field conditions to see whether they repel flies in the field. So in conclusion, at the fruit level, we're finding that spotted wing Drosophila is attracted to volatiles from fruit extracts, but there's differences in the attraction depending on the fruit. So they are highly attracted to raspberry volatiles. We find that domesticated berries are more susceptible and less attractive, and that these domesticated berries are larger and firmer, while wild berries are more sour, sweeter, better defended, and darker. And also we are finding that there's differences in volatile emissions between the domesticated and the wild berries. Also, we're finding that anthracnose infection repels females and also reduces oviposition and immature development. So we're also looking at the chemical ecology of this fly at the plant level. So it is known that females, unmated females, likely utilize fermented odors, leaf odors, and possibly pheromone odors to locate locate host plants and also locate mates. And once mated, they might change their behavior to seek for fruit odors and likely also to yeast odors. So we wanted to see whether these different odors from different sources interact to affect the attraction of a spotted wing Drosophila. So we did a study where we looked at combinations of fermented odors with leaf odors and yeast odors. The idea behind it is that right now we're using fermented odors to attract the spotted wing Drosophila. And we wanted to make these odors more specific to spotted wing Drosophila because other drosophilids are also attracted to this smell. So we wanted to add some other odors from other sources such as leaf and yeast to try to make it more specific. So these are the volatiles that we used that have been identified from fermented odors. These are two compounds that have been identified from yeast that are attractive to a spotted wing. And there's also one compound that has shown to be attractive to a spotted wing from strawberries. So first we looked at the antennal response, whether the insects can detect these compounds in a dose-response manner. And we found that yes, both females and males respond to each of these volatiles, so they can detect them. We also conducted lab studies to look at how they behave towards these compounds, and we found that they were all attractive to a spotted wing Drosophila. So individually, they are attractive, but we wanted to see whether they were also attractive as a blend. So we created a blend of the fermented volatiles, so that's the fermentation lure, a blend with the yeast lure, yeast volatiles, like the yeast lure, and also the leaf lure. And we compare it to a known attractant that was yeast and sugar. So we found that when we look at a larger arena, in these cages, only the fermented lure was attractive to a spotted wing Drosophila, and not the yeast and the leaf odors. But again, what we were interested in was whether when you combine these odors with the fermented lure, does it improve the attraction? So we compared the lure alone and the lure with the yeast, the lure with the leaf, and the lure with the yeast and the leaf lures. However, interestingly, what we didn't expect, we didn't want to happen, was that they were actually they were always more attracted to the fermented lure, and when we added other odors to that lure, we did not see an improved attraction. So we wanted to see whether this happens also in the field. So we created these lures again. These are the same lures, and we put them out in the field. We did that in blackberries, blueberries, and cherries across different states. And we found that during the in-season, only the fermented lure was attractive, and flies were not attracted to the other odors. But they became attractive to the leaf and the yeast odors when after the season was over, so in the off-season. So it's likely that the background noise is competing with these odors in the attraction of flies to the yeast and the leaf. So once there's no leaves and possibly a reduced amount of the yeast, they become more attractive. These odors become more attractive. So we also wanted to see whether, under few conditions, we can improve the attraction to the fermented lure in combination with other odors. And we found the same that we found in the lab, that when we combine the fermented lure with other odors, instead of making them better, we made them worse. So flies were less attracted to the fermented lure when they were combined with leaf and yeast odors. And this was consistent during the season and in the off-season. So clearly we did not combine the right cues to attract spotted wing Drosophila. So next, what we have been looking at is other odor combinations to see whether we can improve their attraction. And one of the ones that we feel like they would be would enhance the attraction, especially to fruit odors, is by adding leaf odors. So we have been doing experiments looking at the fruit when alone and in combination with leaves. And consistently we find that both leaf and fruits are attractive, and also in combination, but when you have the fruit with the leaves, they become more attractive than the fruit alone. And this was consistent also for males. We did this study. We also did oviposition studies to look at whether there were more eggs laid in the presence of the leaves. And we do find the same thing, like females prefer to lay eggs on fruit when you have leaves nearby than when the fruit is alone. But when you give them a no choice, they lay an equal number of eggs. We did also cage studies, and we find the same thing, that the presence of leaves increases the number of eggs that the female is laying in the fruit. but when you have a no choice, they lay equal number of eggs in both. So clearly there's something in this combination with the fruit and the leaves that is attracting more flies. So now we are in the process of identifying the compounds that are present in the leaf and the fruit that are attractive to a spotted wing. Flies also need resources to live, and in general, insects need food sources to survive. And pollen, nectar, and extraflorate nectaries are resources that insects commonly use to live in nature, to survive in nature. However, one resource that has been had not been studied is guttation. And one of the reasons why it had not been studied is because it was thought to be just water, composed of water. However, we found that guttation is composed of more than just water. And we also find that insects are actually visiting and feeding on these guttation droplets, including a spotted wing Drosophila. So we wanted to see how this guttation affects the survival and egg load of a spotted wing Drosophila. So we gave a spotted wing Drosophila different diets that included water as a control, guttation, alone, sucrose, protein, and sucrose and protein. We applied them in paraffin squares, and then we gave this paraffin to the flies. So we found that both guttation and sugar increases the survival of females, and also increases the survival of males. But also we wanted to look at what happens with the egg load. So we gave females, mated females, the same diets, and we dissected the females 1, 3, and 7 days later. So even after 1 day of providing them these diets, we see that there's an effect of guttation and also an effect of sugar and protein on the egg load, on female egg load. But there was a higher separation at 3 days, where guttation, females that fed on guttation had more eggs than the other treatments. And this continued after 7 days. So what we have found so far is that we wanted to improve this fermented lure, but we found that so far the fermented lure is the most attractive to a spotted wing Drosophila. And that combining this fermented lure with yeast and also leaf volatiles did not increase the attraction. However, we're finding that combining fruit with leaf others increases the spotted wing Drosophila attraction. And that guttation increases the survival and egg load of these flies. We have been also studying how the chemical ecology of this fly under in the field. So we did a study similar to the blueberry maggot fly study, where we looked at how management practices and location, trap location within fields affects trap counts. So we did this in 9 different locations, and we looked at what do the trap counts mean? What do they tell us? So again, we looked at, we placed traps throughout different farms in the southern part of New Jersey, and we looked at, and these traps were located either at the border of fields or in the interior of fields, and we also in 1 year collected fruit from these fields and see whether they correlate with trap counts. So these are the data of how insecticide applications affect trap counts. And early in the season, the number of flies in the traps was very low, so we didn't detect any differences. However, as the season progressed, we start to see that insecticide applications does have an effect on the number of flies that you trap in these traps. Also later in the season, we see the same effect. However, in the postseason, when the fly counts start to get too high, we did not see an effect. Also, growers stopped spraying that later in the season. And this was consistent for 2 years. You can see that also in 2017, by the mid-season, you see an effect of insecticide applications on the number of trap counts that you find. So if you apply more insecticides in one field, you're going to see a reduction in the number of flies that you capture in the traps. Later in the season, again, when the counts are high, you start to see more variation and not an effect. We also see an edge effect, a clear edge effect. So traps that were placed near wooded areas consistently capture more flies throughout the season. And this was consistent throughout the 2 years of the study. So there's a more pressure coming from outside, from wooded areas into the fields. And this is consistent throughout the season. And we also find that the trap counts can correlate with fruit infestation. So if you have higher trap counts, there's more probability that you will get infested fruit. And here, in 2 seasons, in the mid-season and the late season, you can see that if you have infested fruits, your probability of having infested fruit in your field increases if your trap counts are higher. So higher trap counts means higher risk of getting infested fruit. So we want to use some of the knowledge that we have gathered in chemical ecology to manage this pest. So we want to develop methods to be able to hopefully reduce the amount of insecticides that we're using in these fields. And one of the ways that chemical ecology can help in developing these tools is by using, for example, strategies that manipulate insect behavior, such as attract and kill. So these attract and kill strategies combine an attractant, a phagostimulant, and an insecticide. And there's some products that have been developed and are under study. None of them are registered so far, but are under development, that include attracticidal spheres that are based on a visual cue, food-grade gum, Combi Protect, and the one that we have been studying the most throughout the years is this splat or hook Spotted Drosophila that combines an attractant, a phagostimulant, and an insecticide. So we have done field studies with small plots and in our center to look at the efficacy of this splat. And we found that this splat can reduce the level of infestation, but it tended to last only about a week. After 2 weeks, it tended to be less effective. We have also done studies under field conditions using modified ATV to apply that splat, and we find that you can get reduction in infestation in commercial farms, but the data tends to be variable and depends on the time of the year. Such as later in the year, we saw an effect. And one of the reasons why we feel there's this variability in how effective this splat is because it depends on the number of flies that you have in these fields. So it can be density dependent. And you can see here we did a study in the cages where we tested different densities of the flies to see how effective this splat was. And when you have high numbers, the efficacy of the splat tended to decline. So one of the reasons why we feel as you see, that these strategies might not work alone in controlling Spotted Drosophila. We feel combining multiple strategies might be more effective in manipulating the behavior of these flies. So now we're looking at using push-pull systems where we combine attractants and repellents to manipulate the fly's behavior. And we're looking at, for example, repellents such as the ones that we're identifying from anthracnose infected fruit to serve as the push component in the system. So we are finding that trap counts reflect management practices at medium-sized Spotted Drosophila populations. We find consistently that trap counts are higher near the wooded areas in the forest habitats, and they can correlate with fruit infestation. And we are also testing products such as plat to manipulate the insect behavior. However, they tend to be density dependent, and combining multiple strategies might work better than just using one strategy to manipulate insect behavior. So finally, I want to finish by looking at showing you some of the work that we have been doing at the landscape level. And the landscape we know that is critical for in the abundance and diversity of natural enemies in your fields. So fields that are near wooded areas tend to have more abundant and more diverse number of natural enemies. However, the composition and the structure of these landscapes can also affect the pests themselves. So you can have these habitats that provide food sources for not just the natural enemies, but also for the pests that you want to target. And this is true for example, the brown marmorated stingbug that is very polyphagous, and also for the blueberry maggot fly that utilizes wild hosts near the commercial blueberry fields. And in a recent study, it was shown that the proximity to this, the percent of semi-natural habitats increase the abundance of exotic pests, but not of those of the native pests. So we looked at how the landscape can affect spotted wing Drosophila in blueberry farms. And for this, what we did was we collected blueberries, wild and cultivated blueberries in pair sites, as we did in the past. We also look at, we place traps in both the wild and the cultivated fields. And also we collected the fruit and look at oviposition. So what we found, this is for males, is that the abundance of males tended to increase in the non-crop habitats later in the season, but also they seem to appear in the traps in the non-crop habitats first. And this was similar to the females. So the female on the traps were higher later in the season, and you can see that at least in some cases at the beginning of the season, you see females showing up on those traps first. When we looked at oviposition of fruit, you can see that the first fruit that was infested by spotted wing Drosophila were from those collected in the wild. So it's clear that these non-crop habitats near wooded fields contain suitable hosts for spotted wing Drosophila, and these blueberry fields are under constant spotted wing Drosophila pressure when they are near these non-crop habitats. And there's now a lot of evidence that proves that this is the case. So just to give you a bigger picture, there is efforts being made to establish cultural control practices against this pest, also that are testing different chemical control methods. And also there's an effort to introduce natural enemies from Asia to control this pest. Also there's some research being done on post-harvest management, but my work has emphasized the importance of monitoring and how we can improve monitoring for this pest, and also on behavioral control of this pest. Moving forward, we're looking at, now there's a permit to release a parasitoid of a spotted wing Drosophila in the US. So we are right now, this is the first year this year where these releases have been made here in New Jersey and other parts of the US. This is Ganaspis brasiliensis. And we want to look at how compatible biological control is with the behavioral control tactics that we have been developing. And this is the work that is being done by my two postdocs, Patricia Prade and Beth Ferguson. So with that, I want to thank my two technicians. Unfortunately, Vera has retired. She retired today. Today was her last day, officially. Yes.
Oh, wow.
And all the funding sources, and you for listening to my talk. So thank you.
chat to see if there's.
Okay.
So I had, sorry, 1 minute. So I know of like at least 2 other invasives that come from Asia specifically, like the Asian longhorn tick and the spotted lanternfly. So do you think it's just imports as to why we have so many invasives from Asia specifically? Or what is your like?
I believe that a lot of it has to do with trade. We just, and it goes both ways. We just published a paper because now Asia is producing more blueberries. We just published a paper in Insects looking at a pest that was brought from the US into South Korea.
Wow.
So this is this midge was introduced also through most likely trade into Asia, and now it has become a pest of blueberries in there. Also, like, so it goes both ways. Like also insects are being moved from here to there as well as from there to here. It's called the blueberry gall midge.
I just had like a follow-up question to that then, like, how are invasives still being like there's a lot of regulations now, especially with like increased like global trade about like invasive animals in general. So why is it that like all of these are kind of just slipping under the radar until it becomes a huge problem for the public?
I think it's just by the number of products that are being shipped. I think it's just a pure, and they can be, as you said, be under the radar. Like a lot of, also people can bring goods unintentionally that are infested. Like it's likely that the spotted wing Drosophila came from different, because spotted wing Drosophila showed up in different parts of the US, and it's thought that they came from different sources. So even you have different introductions, from coming from different parts of the world and being introduced in different parts. And most likely they came from fruit that was infested with the larvae. That just, people bringing in fruits. One interesting thing is like, it was first detected in the West Coast. And my growers were asking me what to do to prevent it from arriving to New Jersey. And that's a very difficult question to answer because we're moving fruits from all over the place. Like when blueberries are not in season here, we're bringing them from South America. Also, we bring them from the southern part of the state. So how do you prevent those that movement of and checking all that fruit that is coming in is very difficult. So so I think we do the best we can, but you're going to see this, these insects that are, are going to establish, yeah.
So we have 2 comments to that question in the chat. Dr. Fonseca says that it could also be similar weather because we're at similar latitudes to where they're coming from. And
That's
then Devin Creedman, who's one of our grad students, comments that there isn't enough inspectors at the ports to catch everything coming through. And he's absolutely right. I've done some work with the Port of Elizabeth, and the amount of material coming in, they just don't have the resources to be able to check everything. And so they do a statistics and sampling program. They only get about maybe 1% of the stuff actually inspected. They do find things, but yeah, they just don't have the funding to do what would need to be done to do that.
And a lot of it also, like the distribution of the pest tends to, like most likely is going to change with global warming. Like you expect that also to affect more areas as, some areas start to warm up. Dr. Hopkins.
Yeah, so you presented data that females were attracted to leaves and fruit,
Yes.
but also that males were too.
Yes.
So is it likely that it's not over-position seeking olfactory cues, but more something?
Yes, I agree. I think it's mostly like looking at the habitat. I think the leaves, what they are doing is just telling the insect that this is the right habitat to where you can find, food and also mates. So that's why I feel like males are also attracted to them. There is differences. These are very hungry flies, so they are always seeking sugars. So that's why they are very attracted to fermented products. And if you put fermented odors versus fruit odors, they always go to fermented odors. They are more attracted to fruit odors if you compare them with Drosophila melanogaster, for example. So they have this attraction to ripening fruit, but if they are always hungry, so they always seek for sources of sugar. And also, this fermented fruit provides them for a site for mating. So males and females, they are always being attracted to this fermented.
So has anyone looked at the difference in responses, like before, like in females, searching for oviposition, so before and after mating, and seeing what is more attractive once
I
they've
don't think,
mated?
no, I don't think we do
Because
that.
I guess these are virgin females that are attracted to the site for food, but maybe then ones that are
But I
mated-
think like the mated also, once they mate, I think they are still seeking sugars. So they are going back and forth, because we find also mated females, because that was our idea that, maybe like we are going to catch more of, like unmated females with the fermented, but no, you also catch them, you also catch mated. I think it's because even the mated ones, they're always seeking also for sugar sources.
So, do they mate multiply, or
I think so,
do
yes.
is it known whether they can
That,
expend their clutch and then feed like some of the parasitoids do and come up develop more, or?
yeah, that I don't think anybody has studied that. That I like Chloe's question, because I don't think we have studied much on that switch. What makes it like change that in the behavior to switch from
Right.
one odor to another.
Because if the oviposition behavior is kind of the key to disrupting this, because it's the egg-laying the larval behavior that's damaging, it'd be interesting to see the differences before and after mating and
Yeah.
see. Probably there's different olfactory cues completely.
I think in a basic as a basic question, I think it's important. In an applied aspect, I don't think is that relevant, because that's what we were trying. Like, I don't see how you can apply it, because we know that they are always seeking for these fermented odors. So I don't know how you can make those odors more specific, or make the fruit odors more attractive.
Yeah.
So that's kind of like where we are right now. So-
Well, I guess if you can just make something that's more attractive to mated females, then they
Exactly,
allude
yeah.
to that before they oviposit.
That's what we're hoping to find.
Yeah.
Yeah, that's exactly. That's our, we have been trying like for 10 years.
So Siga, is
Yes,
that
Chenglu.
so fermentation odor is much stronger, much more attractive than
Yes,
the lower,
always.
the fall compounding or whatever. Very similar to German cockroach. So bread and beer is most attractive than any kind of odor.
Interesting.
We did many experiments. So that means for you, if you want to develop a track and a kill, I guess you just use the fermentation.
That's what people are using, exactly. Those are the older. But the problem is that they are not as specific. It's
Right.
going back to what Chloe is saying. Like, we want to make that more specific, because right now we're attracting a lot of different flies. So to go sort through those traps is very tedious, and growers don't like doing it, because you're attracting not just spotted wing Drosophila, but all the Drosophilids. So we have been hoping to make it more specific. Yes.
Wait, so you said that they were attracted to certain berries over others, and
Yes.
their fermentation odors over others. So,
I don't know about that one.
okay.
Definitely, like if the fruit is healthy, they are definitely more attracted to,
Okay.
to raspberries.
So going with the fermentation question, why wouldn't you just make like a raspberry or strawberry mead?
Because they are always more attracted to the fermented. That's what we were trying.
Well, mead is fermented. It's like a type of like homebrew beer.
The problem, I have problems with just making baits like that, because
Okay.
they don't last as long, and they can with the heat they change their composition. So right now, for example, what we are using is lures, attractive lures, which we're using like the synthetic compounds that were identified from that fermented fruit to attract them. So because they are more reliable, they stay longer. They are manufactured by a company that makes sure that that lure lasts for 4 months, for 4 weeks. So you don't need to be changing. Whereas baits, like in the field, they are going to be changing the attraction very quickly. So you will have to like be rebaiting every week, for example.
Okay.
So that's why we don't go with the fruits in the field.
Okay.
Any other question?
Okay, there are no more questions.
There's one more.
I have two others.
Okay.
I'm
Oh, wow.
sorry.
No, you go.
Pizza's not here yet, so you got time.
These two are actually kind of, I don't know. But so you said like the wild blueberries were more attractive but less susceptible, even though the domesticated blueberries were less attractive but more susceptible. So what is like other than the fact that it's a commercial crop and they'll still be affected, regardless, like what was the overwhelming need? Like why wouldn't you just disperse more wild blueberries within the forest surrounding your field?
So that's a very good point because I'm measuring everything by weight. So by weight, you see that the berries are emitting more, but in the wild, you see less number of berries in the wild than in cultivated, of course. So just by numbers, the fields are going to be more attractive to spotted wing. You see, because you have more berries. So even though by weight that berry doesn't emit as much volatiles as the small berry in the wild, just by the number of berries that you have, that field is going to be more attractive to spotted wing. So they are constantly going to be coming in from the wild into your field just because there's so much food there. Like it's like a, like more abundant as food source for them. Whereas in the wild, you see sporadic fruits here and there. So in the wild, like those volatiles were most likely evolved to attract seed dispersers, like birds, for example, and maybe rodents. So that fruit, that color contrast with the leaf was likely evolved to attract some animals that will disperse the seeds, not to attract the spotted wing Drosophila, of course. But spotted wing Drosophila has adapted to utilize those odors, and they can use those odors if they are there. But again, in the field, you're going to have like a stronger smell just because of the number of fruit that you have.
Okay.
So I got one more in the chat for you.
Yes.
Which one is better for reproduction and development, wild hosts or cultivated?
So for the cultivated is, yes, by far, because they are bigger. They are like at least twice the size as the wild berry. So they lay many more eggs. They can lay like 8 to 12 eggs in a cultivated berry, where they just lay like 1 or 2 or at the most 4 in the wild. And also because the fruit is smaller, there's not enough food for a lot of larvae in there. Whereas the cultivated is a lot bigger. That's why the risk of attack is higher in the cultivated crops, just because the numbers and also they are more susceptible. But that's why the growers have to spray so much pesticides, because they are in constant pressure especially from flies coming from the outside. Yes. This is good.
Let's give our speaker a round of applause.
Okay.
I want to give him a break before we actually
Yeah, you can
answer
stay
more questions.
feel free to stay for.
So,
What do you want to do?
That's my question.
Okay, yes, go ahead.
So for case 3, you said plant provided other resources than just
Yes.
than just the fruit itself.
Yes.
So you looked at gustation. So I don't know
Gustation.
what the results you found for that. That gustation may have also been a factor.
Yeah. So
Your thoughts on that?
So as I mentioned, these flies and other insects, they need to like have, for energy, they have to take some sugars.